So our next speaker here, Dr. Koffi Ouattara, is a lecturer and researcher in environmental microbiology and microbial ecotoxicology at Nangui Abragua University in Abidjan, Côte d'Ivoire. Yeah, how'd I do? It was a whole mix of pronunciations in there. Apologize if I screwed anything up. Well, he is the head of the eDNA laboratory, and in fact, I think the principal person doing eDNA work in Côte d'Ivoire. So a pleasure to have you be able to come with us. He's also the department head of Research, Valorization, Innovation, and Technology. I love the title of the department as well. He holds a PhD from Brussels University in Belgium and has worked on an impressive range of topics, including modeling fecal indicator bacteria in sediment and water in various rivers to look at environmental health, drinking water contamination, environmental impact analysis using eDNA, and more. His talk today is implementing eDNA as a tool for biodiversity conservation in Côte d'Ivoire, opportunities, challenges, and perspectives. Please join me in welcoming more.
Yeah. Thanks to Ryan for this presentation. I hope that I can continue with this good English. Let's go. Before starting, I just want to present myself, what I'm doing, and trying to explain a little bit where I come from, where I'm going. And you see that is something which is very interesting because I start like water engineer first, and then I try to get my PhD water microbiology. And now I'm a teacher researcher since 2012 in Ivory Coast. Normally, when I came back to Ivory Coast, I integrate the laboratory of the environment, and there you have around 30 researchers. Many of people are working on fish and microinvertebrates, and I'm the only one who is working on microorganisms, specifically in bacteria and archaea. And then one day, my big brother, who is the head of the big department of, let's say, this laboratory, said, okay, we have a good project on eDNA, and you are the only one who works on molecular biology. I said, yes, but the problem of what you are explaining is fish and microinvertebrates. I don't know anything like that. He said, normally, if you have a basic information on microbiology, on molecular biology, you can be able to do that. And then I accepted the role of the head of the eDNA laboratory department. Just to say that, okay, we try with, let's say, what we have and what we want to have. Like, we know that with collaboration and relationship, we will be able to, let's say, have a good knowledge and also try to realize what we want. That is why I try to join many structures to have, let's say, more knowledge about the genomics, like Validate Network in UK, for pathogen genomics, Ivorian Microbiology Society. Ecotoxicomics is a French group working on ecotoxicology, and also try to have collaboration and partnerships to be sure that when I have problems, I can ask help. And it's what I learned with eDNA, and I got a problem. They helped me with the good material I received. Just the research I'm doing now. Let's say I would like just to present 6 projects, but for today, I just focus on one of the project. Yes, we start, let's say, the genomic studies with, let's say, a study on the biofilms and pathogens in living water first, and then we try to perform genomics, metagenomics studies, and after that, we receive the good projects of eDNA, funded by CPF. And in this kind of project, we have to collaborate with Nature Metrics, which is the structure in charge of the analysis, and we are in charge of sampling and the extraction. It's the first part of the project. And finally, with an idea of how can I try to have an idea of microbiological, eDNA studies using a laboratory in backpack. I try to develop this idea, and I asked to eDNA collaborative if it is possible. They say yes, potentially. What do you need? I say probably we can have an incubator. It's good for me. And with the material of I have, they sent me this one. I really want to thank eDNA collaborative for that. Okay. And now we are also working on a big project, European project called Atlantico. We are working on the microbiome of ocean and also trying to see the relationship between the microbiome and the microplastics. We have another project with ERB from France, also in the same way. And finally, we try with ERB to try to implement eDNA approach to the West Africa. We have this project called Wamba School. It is a kind of masterclass. We receive students from different countries and trying to implement, to train people how eDNA works, barcoding, meta-barcoding, and also in the last training we are preparing is how to apply the eDNA approach to what we call bioimmunology, like working on the segment DNA. Okay, as I said, I will just want to talk about the project of Bandama, where we start studying on meta-barcoding. Well, why Bandama River? In Ivory Coast, we have 4 major rivers in the country. And the Bandama River is a river which is very particular because we have many activities around the river. In which one of the activities which is very problematic is the mining. You have that's an industrial one, traditional one. And with the traditional ones, you have, let's say, kind of pollution which are discharged in the river. And then population who really explore the potential of the river for fishing, realize that there are some problems with the production of fishes. And then they try to have a good answer of what's happened in the river. And the question is, okay, if you want to protect the river, the first thing we have to do is to know what we have in the river. And the answer is, we don't know anything on it. Or the study we have performed before do not give us the good idea of what can be done, what can be with the whole information of the river. And then we say, okay, why not? We can try to have the information which can be the starting point. But we know that activities are now running. It is not really the starting point, but it can be considered as and what we are trying to do. It is like just to show you how it works, like it is, we have a kind of river with this kind of color. Because all activities are putting the, let's say, the pollution on the river. Well, before trying to implement our studies, we say, okay, what we have as, let's say, the basic data, what have been done with the traditional methods. In 2008 and 2009, we realized that people were working with the method of electrofish and also trying to have, let's say, a kind of capture of fish emerge, have, let's say, a kind of information on the fish they found in the middle, the river. This is the first information we got from the 10 years or the 10 past years. The second one studies a study who focused on normally the rivers start in the north of the Ivory Coast, and in the south of Africa, south of the Ivory Coast, you have, let's say, kind of estuary. And in this part of estuary is very interesting to study. And there are some researchers from Ivory Coast who try to understand what happened here in terms of biodiversity. And also, I try to see what they have as diversity they found there, to be sure that our studies, when we start from the north to the south, we can have, let's say, a global information about what have been done, what are the critical points, and what should be, let's say, a good thing to do. With this project, we start in March 2021. or to 2022, 2 hours, two years. And then we have, let's say, around 45 sampling sites. We select 35 points along the river and 10 extra points, which are sometimes points with the tributaries coming to the river. And we try to select with our partner from Nutrimetrics what region we want to work on. And they use Illumina sequencing to do that. And for results, we focus on the O2, similarity around 99% for species detection and 97% for kinase. And we try to have this information about using the database of the GenMate. And the result we got is like we have around 4 families and 19 other orders. Well, in this study, we realized that the information we got do not help us to have the name of the species. We just focus on the family and the orders. And what would be a good question to ask is why we cannot go to the species? And we realize that we have a problem of database. And then we said, okay, with this, we can try to have, let's say, the information of the different family we have, different order we have, and try to see, it's not a comparison, to see what people have done in the past. Is there a change or not? And you can see that for the 2 studies I showed before, the group of persicum are the dominant one. And when we realize that all this family or special orders we found in our eDNA studies are quite similar to what has been done in the past in terms of, let's say, classical methods. But what is really interesting is with this eDNA approach, we found that there are some specific, let's say, family we do not found with, let's say, the classical method. It's not a comparison, because to have a good comparison, we have to do the same work at the same time. But just to have an idea of like what we are doing, we are in the right way or not, is our idea. This is what I try to do, is like for the estuary parts, we have more information on this part, and we select the same point. So like, can we have an idea of what they have found in the past and what we have now? And when you realize that you have, let's say, there are very different between the normal family, but we have, let's say, quite similar families we found there. In terms of order, it's the same order. And clearly, when you use the fishing or the electrofish, or let's say the capture, you can go to the genera or go to the fish species. But in our studies, it's not possible now. That's, I think, this can be the black points in our studies. We can be correct if we have, let's say, the good tools to analyze our data. It's good. And what is really interesting is when we are working with the classical approach, it's sometimes difficult to have an idea of what are the species we have to protect. But with eDNA, it's not simple to have this information, because you found this species and know that these species are, let's say, in the reference of the endangered species in the red list. And for us, it is like we know that we have now some species we have to focus on. Probably we have to organize now our sampling campaign to be sure that we can have more information about the species we found that there is a problem for that. And in the scope of the project too, we have a part of sensibilization and training. We realize that in the project, we have to send the filters to UK for the rest of the process. And we saw that, we realized that also that it will be very difficult for us to convince our group of research who works only with the traditional approach that we have to move on the new one. And then we asked to change one part of the project to build a kind of small laboratory in Ivory Coast. That's why we try with the overcollect to perform a barcoding. I will show you the result of the laboratory. That is what I call challenge. The laboratory is like this one, the small one too, and a really small one. And we have, let's say, this kind of equipment to start the work, like iron, laptop, small equipment, and the bento lab for, let's say, the amplification, a continuous parameter for, let's say, the quantity of eDNA, and the incubator line we use it for the process of, let's say, amplification. And then we decide to take 6 fish from our country. We know the name, like Côte, Zili, and Ouattara, and say, okay, can we be able, with our new tools, to find the same names of the fish, and then to prove to our colleagues that what we are doing, what we are saying, have sense. And you know, in my country, like this, if it's work, you have to do that. And I think they are right. Because they are working always with the traditional method, and you want to change the paradigm. If you want to change the paradigm, it's like you need to have good arguments. And you try with this one, and yes, we test the 6 fish. I can say 14 year opportunity. We just find 3 of them in the We just find 3 of them in the database. And we also realize that 3 of the species we know are not referenced in the database we use for the gene bank. And also we are limited to say exactly what type of species we have as, let's say, to be complete in the identification. Clearly, this shows us that we have a kind of gap of what we want to do and what we can do. You want to identify the fish, and these fish are not, the basic one are not in the database. It means the first job you have to do, it is not to implement eDNA. It is to try to have, let's say, a good database reference first. And then you can say, if I have a sequence, I can try to identify correctly species I'm talking about. And just talk about the opportunities. When we talk to our colleagues that with one water sample, we can give information about fish, macrovertebrates, and all bacteria. And we can do it in 2 days. I think how it works. Because normally, in the traditional way, we have a team to go to the field. And we spend 1 week, 2 weeks in the field to have information in the explore. And we try to present the results. I said, me, I'm a microbiologist. I'm trying to give you information about fish, about pests, media, amphibia, reptilia. Exactly, you get some meat.
Can we go back?
It is not like I know what I'm talking about when I'm talking about reptilia, mammalia, but just to show them that with what I'm talking about, we can have a good result. And then if they want to do what we are trying to implement now, we can have, let's say, a good result for our system. And then now I think many people of my lab agree with the idea. And what I realized too is like, they said in my village, when you are the only one who have a school degree, don't be surprised that you have to read the letter of all the people of the village. Now I say, like, we want to do this, you want to do that, you want to do this. And I realized that I'm the only one who have to manage all these things. But I also realized that I'm lucky. Because like a round of climate, I'm the only one who have one thing. That's good. Then I said, okay, I will try to have a strategy there. with the strategy, we are going to implement progressively the eDNA approach, but not to change what we are doing in the past, just to complete what we have now. We have let's say the whole one approach, and we are going to progressively implement the new one, and then see how they can match sometime together, or how this can complete the other ones. Clearly, in my lab, we are making a reflection on three things: barcoding, meta-barcoding, and metagenomics. Barcoding for strategy of conservation, meta-barcoding for biodiversity and patient dynamics, metagenomics for development of biotechnology. Because we know that the two first points, we have an interest for people who are working on the park and let's say natural reserve. They want to have, let's say, good knowledge of the diversity they have in the park or something like that. And then, as all complexity of science, we need money. And if you have good partners there, you can implement your study. And for metagenomics, we decide to implement this one because we also realize that many structures are interested by the results or the byproducts from genomics, in particular for bacteria. And we say, okay, if you have this kind of, let's say, approach, you can try to have more projects and then you can implement the rest of the eDNA approach we want to do. And it is probably a dream, but we want to dream. Yes. I asked to Megan yesterday, I want to say something. Is having a lab for the field. When I visit your laboratory, I realized that there are many things I need, but I don't have. How can I manage the small equipment I have to be sure that I can have, let's say, a kind of acceptable result in the fields? Because it is really challenging in my country. We don't have space. I say space in my university, you can't have space to have a laboratory, a big laboratory now. You have to imagine the space, a small space where you can work. And when you have to convince the park managers, they want to have someone who can do something quickly in the same space. And we realize that this can be something we can try to implement there. Portable genomics to do barcoding and meta-barcoding. It means like something we can try to make people involved. They are not all scientists, but when we saw that, we realized that we are doing something which is very simple for them, it can be acceptable for them. This is the idea. Okay?
Oh, did that not go?
Good. I know this place. Now I'm here, trying to have knowledge. And I know that knowledge without experience, I'm here in Seattle today to have experience. Benefits of your experience, be sure that one day I can create something. And the thing I want to create is to lab for the people. Possible or not, I want to try. And I believe that is possible. Yes, I can say all for me. I don't know if you have something like that.
Yes.
Yeah. Okay. Just to thank you. Me in the field. Me trying as a one-eyed man to help blind people. And me in the all of my, let's say, classroom, trying to explain how mineral works. And what is really interesting in this story, like when I start with my colleague from UK, we perform all the experience in the lab, and people say, okay, this is something which is really exceptional. They came, and then in the lab, the role, we say, okay, we are going to make a demonstration to show that what we are doing is really interesting. And we realize that with the mineral, it doesn't work. Okay, it's okay. We are going to change. We are going to take a movie and try to explain its words. Many things.
So you mentioned that one of the challenges is not having sequences available for some of the species in your reference database and in publicly available databases. And this is obviously something that a lot of us are coming up against with eDNA projects all over the world and through different taxa. Are you doing some barcode sequencing to try and increase representation? Like, do you have the ability to get tissue samples from some of those species so that you can build out your reference database?
Yes, in the scope of the project, we perform barcoding for let's say around 20-25 species. And for the freshwater, we test 6 fish. And I also try to do it for the marine fish, 10 fish. But the problem is now I have to sequence all of this to have the information. And I realized this morning that if we want to have the good information, we have to focus on the specific region. Be sure that we can have, let's say, the information can be used by the people. Because I asked the question of the COI, the 16 gen, and also the 12S. And I know that in, let's say, many people I'm working with focus on the COI. When I ask, they say, okay, it is because they use birds. And with birds, it's more simple to use the COI. But I'm not sure that we have the same information about it as well as babies. Clearly, I think that it is something I'm trying to have a good approach. And I also asked to an experiment person in the University of Côte, who said, okay, they have a good project on vertical geoscan. And she said, okay, if you are interested, we have a proposal for that. And you can see how we can help you to have a second database for you. And it's local, and then you can use it to improve the result. Because we have the same species in West Africa. If you have a good database, it can be very interesting for the rest of the West Africa. Some ideas. And I also asked them, is it possible to have, let's say, a database to test what I have? Probably I can test in this week, and next week. Good. Yeah. Good. That's okay.
I'm just curious, how many or how percentage of the endemic species that you have for this data? Is it like the common one in Africa, or do you have the endemic one from this?
Difficult to say we have endemic one, because the difficulty is not difficult, because the reality is all the major rivers we have are connected to 3, 4, 5 countries.
Oh, okay.
What we call transboundaries rivers, something like that. Yes, it's like we have, let's say, a connection with other countries. It's like possibility to have the same fish in the river. Yeah.
And do you know how many percentage from the total that you already described?
No.
So it is more.
I think more than this one.
Okay.
I think more than this one.
Yeah, because I just realized you always told me about the river from yesterday. I just realized it's like a huge rivers.
Yeah.
So I'm thinking about rivers like a small one.
Yeah.
That's like a huge one.
I don't know how to say it in English. In French, we say fleuve. It is different from river.
Okay.
Yeah.
We have, let's say in French, we say rivière,
Stream.
fleuve, océan. It's the continuum. But in English, I don't know how to say fleuve. When you talk about fleuve, they say river.
That's
Yeah.
a different.
Exactly. So in my country, we say river for small one,
Right.
fleuve for the big one. And this one is really a big one.
It's a really,
Yeah,
really
it's a
big river.
big one, exactly.
It's a really big river.
Yeah, exactly. I can say small river and big river.
Yeah.
This one is a big river. Yeah. The small one. It's a very big river.
Well, I think I'll ask Zach asked Jadita, I'll ask you the same question, that if, what do you think is the limiting factor in getting eDNA into acceptance, into routine use? And is it trainings? Is it, I mean, funding is always an issue. Oh no, I think it was it Canada wants that question. Yeah.
Probably it's the same. I think the best thing to do is training first. Because people are they have it. They are doing things the same one each time. And if you want to change this, clear that you have to prove that you can have good answer or have let's say a specific answer that they cannot have, they don't have. Like for example, when I talk about bacteria, the people in my lab work with the classical method, culture-based method. And when you say, for example, I am planning to do metagenomics, have, let's say, the microbial profile, not identification, but profile, something which is new. With the culture-based method, you cannot do that. For the fish, it is the same. We are always trying to use barcoding or meta-barcoding for identification. For me, we have to go far from this one, probably to try to see if there are something like some information which can help to understand the physiology of fish, for example. This can be something new. For a classical taxonomy one, he take his fish, okay, the eyes like this, the mouth like this, but you can't say anything about the physiology. But with eDNA, I think normally if you have more information about the DNA, you can try to have good answer about this one. It's something I think we can try. And probably also for mutation. If you have something like we change, I think with eDNA you can have a good answer for that. For me, it is first training, and after training, of course, the form to test, let's see our hypothesis. Because if you say something and you don't have the possibility to justify this one, I'm not sure that people can accept. Yeah. Probably.
No, I was going to ask more about the Wamba school. You said that there's eDNA training happening. Are you involved in that? Is it all the way from collection to bioinformatics, or is it just focused on one piece? Or
in the projects, we associate two different views. The first view is the people who are working with the classical taxonomy, and our group working with eDNA. And then we do the first exercise, which is, let's say, the morphological identification first, and then we perform barcoding. And what we plan to do normally is to start with the extraction of DNA to the bioinformatics.
okay,
But the problem is that too many students, they will have macromaterials, each of them, be sure that they can, let's say, do it themselves. What I'm trying to make as a reflection now is how can I try to get them involved, even if I use something possible, something like that, to be sure that you do it, but it's not like I have, for example, the fungus, the old ones. I know that that's works, and they can use this kind of things to have, let's say, a kind of first experiment with this one. Right? Because I think that when people do something, more simple for them to accept it. It is the idea now. We are trying to make a reflection on it, and probably I think that for the next, we have the possibility to make a very important training, because they are coming from different countries, and some people have a good background, some other ones good background, molecular, and have to deal with this different type of people. There are people who are managers, back managers, who are really students, and we have sometimes people coming from the survey of fish. We want to understand how it works, how we can implement this, and it is very interesting. This we have done this next year, in the second edition this year, and probably I think each year we try to address how we can improve this one. And I think for the next years, we can have a good model of learning, which we can do the whole process. Okay, that's for mathematics. Really challenging.
Yeah, very challenging.
Exactly.
Anything from online? Great.
Good. You're always ready for a snack.
Thank you.
Okay, well, I think please join me in thanking Nouho for and really for to both of you, Dita and Nouho, thank you again for coming. And we can say goodbye to people online, and for the people here, we can go have a drink, have a snack down the hall, and continue on. So conversation will go on, and thanks again.
All right. Thanks, everybody online. We are shutting you off. Bye.